Okay, so this is our third little video in conservation biology, and this one is looking at populations and genetic diversity and the Hardy-Weinberg principle. So we've already talked about populations, and I said that populations are very important, and they're very important because they are the arena in which genetic material acts. So a population is probably the most important unit which you need to conserve. This is an example of what a population is. This is the Ethiopian wolf, so this is Ethiopia, and you can see that the populations occur in patches spread across the highlands of Ethiopia there. So each one of these little patches are a more or less discrete unit or population of wolves. So a large one may have a lot of wolves and a smaller one will have a smaller number of wolves. But because they're separated quite often by these valleys and so on, the genes don't often travel between one population and the next population, so they tend to be relatively separated. So the genetic material in one population tends to stay in that population, unless you get one individual wolf migrating across the valleys in between and transferring genetic material from one population to the other. So populations are the units of evolution. And what this means is that the population is where the genetic material acts. So a population has its reserves of genetic material, and when the environment changes, it's that genetic material that the population must draw on if it is to adapt and change to suit that change in environmental conditions and so on. So a population is really what you need to save if you are going to try and conserve a particular species. So protecting population and their inherent genetic diversity is the key to preserving species. If you don't protect the populations, then you're not going to save that species. So what is a population? A community of actually or potentially interbreeding individuals of the same species. It's probably not a good idea to use the term community because that implies a certain taxa altogether, but a population is all of the same species and therefore quite often interbreeding. They share a common gene pool. So all the allelic diversity, genetic diversity within that population is available to future generations of that species. Populations exist within a limited geographical area, and as I said, they are the units of evolution. So the future generations of that species within that population have access to all of the genetic material within the population, so that the genetic material which is most suited to the environment should be the ones which are selected for and go forward. So populations are characterized by alleles in the gene pool. So a gene pool is the collection of all the genetic diversity within that population. So it's a complete set of genetic information contained within all individuals in that population. So a nice little graphic to reinforce that, all these doggies, and this is a gene pool, and those are all the genes that you can see floating around in there, and all of those genes are expressed in the individuals which make up that population. So the gene pool refers to the sum of all genetic variation and represents the inherent ability of the population to adapt, evolve, and survive in a changing environment. pretty much what we were talking about earlier on. So, how does Hardy-Weinberg get into this? So Hardy-Weinberg were a couple of scientists who research population genetics, and they came up with the concept of Hardy-Weinberg equilibrium. Under a population which is under Hardy-Weinberg equilibrium remains constant. So the genetic makeup of that population remains constant, generation after generation. The same alleles are present in the gene pool, generation after generation, and the proportion of those alleles remains the same, generation after generation. And a population is said to be under Hardy-Weinberg equilibrium if that is the case. So there are certain strict conditions which apply if a Hardy-Weinberg equilibrium is to be observed in a population. The first one of that is random mating pattern. That means that every individual in the population has an equal chance of mating with every other individual in the population, and that means that the alleles in the population have an equal chance of making it to the next generation. So all alleles in the population have an equal chance of making it into the next generation. So that means there must be no selective forces, no evolutionary forces changing the genetic diversity in the population or the allele composition within the population. So for that to happen, there must be a large population size, which would mean that random genetic drift does not occur. Now, you don't know what a random genetic drift is yet, and we'll talk about that in the next video, I think. You must have isolated populations, and there is no gene flow between those populations. So if the genetic makeup of a population is going to be the same generation after generation, there can be no new alleles entering into the population from outside through migration. There is no selective forces, so the environment does not act on the population to select some alleles over other alleles. Now, this is fairly rare. I would say that practically every population is going to have some selective forces acting on it. And finally, there is no mutational disequilibrium. In other words, theoretically there is no mutations occurring in that population which introduces new alleles into the population. Okay, so under Hardy-Weinberg equilibriums, there is a large population size, so there is no random genetic drift, which means a loss in alleles over time. The populations are isolated, so no new alleles are introduced. There are no selective forces acting on the population which favor some alleles over the other and therefore changes their proportion and even eliminates some alleles from the population. There are none of that. And there is no new alleles being added through mutational disequilibrium. Also, there is non-overlapping generation. So to keep things nice and discrete, each generation produces the next generation and then no longer participates in any of the future breeding or contributions to the next generation. So that is okay in short-lived organisms, but in long-lived organisms, obviously, that will tend to be violated. And finally, under Hardy-Weinberg equilibrium, there must be sexual reproduction and there must be diploid segregation. So diploid segregation basically means that each organism carries 2 copies of the gene. So they can be heterozygous. They can have 2 alleles in the one organism. Sexual reproduction means that recombination can occur, because it is through sexual reproduction that you get recombination and shuffling of the genes so that different alleles can be brought together in a random sort of way. So sexual reproduction must occur and diploid segregation must occur to meet Hardy-Weinberg equilibrium conditions. So if all of these conditions are met, you will be able to get a frequency of, or you'll be able to calculate or be able to assume that each allele has a certain percentage chance of reaching into the next generation, and this is what all these figures indicate. So if you have one gene, or one locus, which has two alleles, okay, in a diploid organism, that would mean there would be 3 genotypes. Okay, so the genotypes would be, if the alleles are there are 2 alleles at a locus, capital A and little a, that means there can be 3 combinations of capital A and little a. There can be 2 capital A's, 2 little a's, and 1 capital A and 1 little a. So which one of these genotypes is the heteromorphic genotype at this locus? Yes, correct. The capital A and the little a genotype is the heteromorphic one. So the higher proportion of this one we have, the greater the genetic variability there is in the population. So the higher proportion of individuals with this heterozygous genotype, the greater the genetic variation in the population. Alright, so genotypic frequencies are associated with individuals in a population. So the frequency of AA can be, say, denoted as P, frequency of the heterozygote is H, and the frequency of the homozygous to small a is Q. And these are then associated with the gametes which are available to the next generation in the transfer into the next generation's gene pool. So the frequency of gamete A would be P, and the frequency of gamete little a would be Q, and if you add those together it will give you L. I'm not really too sure why, but there you go. So between genotypic frequencies and allelic frequencies, you would find that the frequency of big A, which we've denoted as P, would be equal to the number of homozygous individuals plus half of the heterozygous individuals. I mean, that makes sense, doesn't it? The number of, or the frequency of, say, sperm or eggs with just the big A. Remember, sperm and eggs are going to be haploid, and once they join together, then they become diploid. So the number of gametes with big A would be all of the big A individuals, because they're going to have all big A sperm and all big A eggs, plus half of the heterozygous individuals, because only half of the sperm and only half of the eggs are going to be big A. The other half of the sperm and the eggs are going to be little a. Okay, so the frequency of this particular allele, big A, is going to be proportional to the number of individuals which are homozygous to big A, plus half the individuals which are heterozygous with big A and small a. Okay, so that will give you the allelic frequency within that population of, say, big A, and you can work out the allelic frequency of little a similarly. Okay, and that's interesting and important because it gives you an idea of how much variability is going to be inherited from one generation to the next. Okay, so the probability of the next generation being a heterozygous looks to be, fairly decent, I guess. I'm not going to say anything more about that. I'm really not too sure what I should say. But basically, at Hardy-Weinberg equilibrium, the allelic frequency of the population will remain constant. So that means that if the allelic frequency is directly related to the genotypic frequency, then the genotypic frequency must remain constant as well, generation after generation. So if you are finding that your genotypic frequency changes from one generation to the next, that means that the allelic frequency is going to be different, and therefore the population is no longer at Hardy-Weinberg equilibrium. So allelic frequencies and genotypic frequencies are going to remain constant generation after generation if a population is at Hardy-Weinberg equilibrium. And this is the principle of conservation of gene frequency, and it means that a population is not losing genetic diversity, and that means it's going to last into the future. Also at a Hardy-Weinberg equilibrium, there's a direct relationship between allelic frequencies and genotypic frequencies. Well, this is pretty much what we set up here, according to this relationship. Okay, so it's easy enough to measure genotypic frequencies if you have a number of individuals, and say for instance you can measure, say for birds, wing length or something like that. You can work out the frequency of birds with long wings, for instance, and if that changes over time, then we know that the population is not at Hardy-Weinberg equilibrium, and therefore there must be something going on there. Okay? Alright, so what do we know from this video? Populations are the units of genetic diversity. They are the units at which genetic processes occur, and therefore the units at which evolution occurs. So we need to pay strong attention to populations when we try and conserve genetic diversity, and also to conserve the species. The Hardy-Weinberg principle is a baseline, and it gives the conditions for a population at which genetic diversity does not change. And we should try and be aiming for a Hardy-Weinberg population if we are looking to conserve, at least in the short term. In the long term, there's probably selective pressures which is going to change things, but at least in the short term, if we see a population which is departing from the Hardy-Weinberg equilibrium drastically, there is probably something wrong which needs to be fixed up. And quite often it's the small populations which are not at Hardy-Weinberg equilibrium, and that's what we're going to talk about in the next video: changes in population genetic diversity. In other words, populations which depart from Hardy-Weinberg equilibrium, which is basically all populations, but particularly small populations which can depart very quickly from Hardy-Weinberg equilibrium, and we need to do something about that.